Hello friends, welcome to BMH Learning. In this video, we are going to discuss about estimation of glucose by Cole's method. Introduction. Reducing sugars have got free aldehyde and keto groups. When boiled, these groups have the ability to reduce metal ions such as ferricyanide ions under alkaline conditions. Apart from this, the reducing sugars can also reduce the redox indicator dyes such as methylene blue. Principle. When boiled under alkaline conditions, reducing sugars undergo tautomerization. For example, we can talk about glucose, which is a reducing sugar. So, when glucose is boiled under the effect of sodium hydroxide, the sugar undergo tautomerization at first and second carbon to form 1,2-enediol, which is further tautomerized to enol salt. Now, 1,2-enediol is actually a very powerful reducing agent. That's what is important for us in Cole's method. So, when there are hot alkaline conditions present, the glucose can reduce potassium ferricyanide to potassium ferrocyanide. Because it's a redox reaction, something must be oxidized. So, glucose is oxidized to form a complex mixture of organic acids. Then, to confirm the redox reaction, methylene blue is used as redox indicator. Before the reaction, the dye is in oxidized form, and with the reducing sugar glucose, it gets converted to its reduced form called leuco compound. So, once all the molecules of ferricyanide are reduced to ferrocyanide, the end point is marked by a color change from blue to colorless. Requirements. We need 1% potassium ferricyanide solution and 2.5 normal sodium hydroxide. So, 20 ml of potassium ferricyanide is mixed with 5 ml of 2.5 normal sodium hydroxide to get a yellow colored solution. We prepare 3 flasks containing such yellow colored 25 ml solution. These can be named as PR1, PR2 and PR3. PR here stands for pilot reading. We are naming the flask this way because we are gonna take 3 pilot readings. Pilot reading 1. So, PR1 is kept on a burner for boiling. Few glass beads are added so that bumping doesn't occur during boiling. Then 1 to 2 drops of 1% methylene blue are added. Addition of methylene changes the solution color from yellow to green. After this, using a 1 ml pipette, add glucose sample solution into the boiling mixture. As we keep on adding glucose drops to the PR1, the color changes from green to violet. We need to continue the titration until the green color disappears and the solution becomes colorless. We should stop here adding glucose because this is the end point of titration for PR1. Note down the volume of glucose solution required to achieve the end point. Record the value as pilot reading 1. Repeat the same procedure with PR2 and PR3 as done with PR1 and record the values as pilot reading 2 and 3. Now let's suppose the pilot readings are in this fashion. PR1 is 1.2 ml, PR2 is 1 ml, and similarly PR3 is 1.1 ml. So the mean value we would be getting is of course 1.1 ml. This is called as p-value. Now we have to dilute the original glucose solution in a way that the final titration ends at volume of 10 ml actually. So if we closely look at the variations in these PR values, we must assume that the final value must fall between 9 to 11 ml. So in order to make final 10 ml dilution, or we can say the volume of glucose sample, we need the average, that is 1.1 ml of glucose, and we require 8.9 ml of distilled water to make it 10 ml. Now at least we require 3 final readings, and assuming any chances of experimental error and loss of solution, we need 40 ml of diluted glucose sample. So that means the amount of original glucose sample is going to be 4 times. That is, the p-value, which is 1.1 ml, is multiplied by 4. This gives us 4.4 ml. And the volume of distilled water to make final volume 40 is 8.9 into 4, which is 35.6 ml. So 35.6 ml of distilled water plus 4.4 ml of glucose sample give us 40 ml of final volumes to make 4 10 ml volumes to take final readings. So what do? We add 4.4 ml of original glucose sample to make 40 ml diluted glucose solution, which is more than enough to make 3 final readings. Let's proceed to final readings. Prepare yellow colored solution in same volume as we did earlier by mixing potassium ferricyanide with water to make 25 ml. Name it FR1. Similarly, make other solutions and name them as FR2 and FR3. All the 3 final readings are taken in similar fashion, that is, in the way we recorded the values for pilot readings. The difference is that in case of final readings, we will be using at least around 9 to 11 ml of diluted glucose solution with 10 ml pipette. Now let's suppose these are the final volumes of glucose solution. The mean value is 10.1, and this is called as X value. So now we have both readings, that is P value and X value. Put the values in the formula and you will get the result. That's all. Thanks for watching.